Welcome to Wildcat Science with Mrs. Haugo. This is Chapter 9 on the nervous system, Part B. We are talking about types of neurons. There are sensory neurons or afferent neurons with an A. These neurons send messages to the spinal cord or the brain, so they go from the body to the brain. For example, if you push on your finger, you feel that because sensory neurons are sending a message to your brain. The opposite, or a different neuron I should say, are motor neurons or efferent neurons with an E. These carry impulses from the spinal cord and the brain to the muscles and the glands. And they do something. For example, your brain is telling your muscle to contract. The third type are interneurons, and they relay information from one neuron to another. So let's look at a picture of it. Okay, starting up here in blue, we have our sensory neuron or afferent neuron, and it is getting signals, sensory signals, perhaps like pressure from your finger, and it's sending it down the neuron into the interneuron, which relays information from one neuron to another, to the motor neuron or the efferent neuron, which tells it to do something. Perhaps move your knee. If you notice, this is the spinal cord, so this information didn't go to the brain, so maybe this is a reflex, like when you hit just below your knee and your knee kicks out. So nerves are like neurons, but a bit different because neuron fibers are collected into bundles called nerves. So a neuron is an individual cell, whereas a group of neurons form a nerve. So like a neuron would be the cell and the nerve would be the tissue. There are three types of nerves. Afferent nerves, which carry sensory information from the body to the brain, like pressure, temperature outside, the temperature of your skin, and pain. Efferent nerves carry motor information from the brain to the body to respond. And then we have mixed nerves, which carry sensory information from the body to the central nervous system, and they contain motor neurons carrying information from the central nervous system to the body. So if we look here, these are our individual neurons and they make up a nerve, and this could be a mixed nerve, and so some neurons could be sensory and some neurons could be motor, but they don't mix. One will be sensory and one will be motor. And then they're both in the same nerve. So we've got axons, myelin sheath, connective tissue, blood vessels, all that good stuff in the nerve. Afferent and efferent can be pretty confusing. Let's look at the words. So it helps if you remember the difference between the English words affect and effect. The music affected her deeply. Something is experienced, some sensory information is experienced, and this is your afferent neurons or nerves pick up sensory information and send it to the brain. His protests had no effect. Something is done or not done, and that is a motor output. The efferent neurons or nerves carry impulses from the brain and make the body do something. Let's look at that in a picture. We have sensory receptors in our eye, and then afferent neurons take sensory information and bring it to the brain where integration happens. And then there is a motor output through efferent neurons, which looks like you're contracting a muscle. Perhaps this person saw something and is now running away. Let's talk about neuroglia. This is another type of nerve cell. So neuroglia, or glial cells, are another type of nervous cell, and they are basically the support crew to the neurons. and it means nerve glue, neuroglia. So they protect and nourish neurons. They support nervous tissue and bind it to other structures, and lots more jobs that are essential to you. Different than neurons, the glial cells or neuroglia divide throughout their life, and they're actually much more numerous than the neurons. They outnumber neurons 10 to 1. Okay, let's take a look at some of our other neuroglial cells. You got a little sneak peek before. And let's talk about the astrocytes. These are star-shaped and pretty numerous, and they bind nervous tissue to other structures like blood vessels, and they are involved in metabolism and synapse formation. Another type of neuroglial cell are microglia, and they act as phagocytes, and they just pick up foreign invaders so that they don't infect neurons. You have oligodendrocytes, which produce the myelin sheath in the central nervous system. And you have satellite cells that surround the neuron cell body and protect it in the peripheral nervous system. So let's talk about the myelin sheath, which is a very important part of nervous tissue. So some axons are covered in a fatty material called myelin. Myelin insulates and protects the axons. Myelin also increases the speed of electrical impulses, which is the message that's trying to move down the neuron. So faster is better. So myelin is made of Ch9 cells, which are a type of neuroglial cell. And the spaces between the different Ch9 cells are called the nodes of Ranvier. And the electrical impulse jumps from one node to another. In a disorder, we're going to learn more about multiple sclerosis, or MS. Patches of the myelin are destroyed in the brain and the spinal cord. And as we know, it increases the speed of electrical impulse. So this means those impulses are slowed down, and it affects movements. So let's take a look at some of these myelin sheath. As you can see, we have our neuron with the cell body and the dendrites, and then its axon, and then this fatty material made out of Ch9 cells is the myelin sheath, and then there are nodes or spaces between the different Ch9 cells, and these are the nodes of Ranvier, and the electrical impulse actually jumps from here to here to here, 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 instead of having to go all the way through. As you can see, the myelin sheath makes it a lot faster. And here's another picture, and then this is in the central nervous system in your brain. Okay, brain teaser time. So Newville is a town where 5% of all the people living there have unlisted phone numbers. If you selected 100 names at random from the town's phone directory, on average, how many of these people would have unlisted phone numbers? Take a moment and write your answer down. 0. See if you can figure that out, and we'll discuss it in class. So let's talk about the nervous system at work. The nervous system works by sending electrical impulses along neuron fibers and transmitting chemical messages from cell to cell. So electrical signal along the neuron fiber, and then to get to another cell, it sends a chemical messenger. So nerve impulses travel down a neuron fiber like a wave. And a nerve impulse is transmitted when a resting neuron is stimulated and undergoes an electrical change. So a nerve impulse happens when a resting neuron changes. So let's talk about a resting neuron. What would a neuron at resting state look like? Well, first we need to talk about the plasma membrane of a neuron, because it is more negative inside the cell than outside of the cell. This difference in charges, called an electrical potential, will be used to do work. Just like if you have a battery which has a positive and a negative end, that is electrical potential, a difference in electrical potential, they are used to do work. Your electrical potential on each side of the plasma membrane in your neuron can be used to do work. Also, the concentrations of sodium ions, Na+, and potassium ions, K+, are important. Sodium potassium pumps are busy sending sodium ions out of the cell and pumping potassium ions into the cell. And then this concentration gradient of a lot of sodium on the outside, a lot of potassium on the inside is going to be used to do work. So what is that work that needs to get done? Well, let's talk about a nerve impulse first. So a nerve impulse starts with a local reversal in the plasma membrane's electrical potential. So we talked about it being negative on the inside, positive on the outside. So a reversal would be where it gets positive on the inside and negative on the outside. And when we say local, it just happens in a little spot, and then this spot travels all the way down the axon. So this reversal in electrical potential is caused by the movement of ions, in particular the sodium and the potassium ions. A sudden electrical change in the membrane is called an action potential. So let's talk about an action potential. In an action potential, there are 3 or 4 stages. The first stage is resting, where the neuron is at resting state. And we talked about this previously. The cell is more negative inside than outside, and there are more sodium ions outside the axon, the cell membrane of the axon, and more potassium ions inside the cell membrane of the axon. Also, voltage-gated membrane channels in the cell membrane are closed. So membrane channels, these are proteins in the cell membrane, and they are voltage-gated, which means a change in charge, positive or negative, will open them up. The second stage is depolarization, because at resting state, we say that the axon is polarized, or the cell membrane is polarized. So to depolarize it, we're going to switch charges. So a stimulus, this could be electrical, chemical, or mechanical, opens the voltage-gated sodium channels and sodium ions rush in. And we know that they're positive, the sodium ions, so the cell becomes positive on the inside and negative on the outside, and then those voltage-gated sodium channels close. They're only open for a little bit. And then the cell will repolarize. The third stage is repolarization, where the membrane becomes negative on the inside once again. So the voltage-gated potassium channels, they're a little slow on the uptake, and they finally open, and potassium ions rush out of the cell. And so the cell returns to being positive on the outside and negative on the inside, because we had a bunch of positives on the inside that now flow out, and now it's positive on the outside and more negative on the inside. The fourth and perhaps first stage is resting. So the sodium-potassium pumps get to work and restore the sodium ion and potassium ion concentrations. So locally, the membrane potential is back to normal, where it's negative on the inside, positive on the outside, and then we have sodium ions on the outside and potassium ions on the inside. But elsewhere in our neuron, along the axon, the sodium ions have moved forward along the inside of the axon and caused the voltage-gated sodium channels to open up there. And then at that new spot, the whole process of depolarization and repolarization happens again. And this repeats itself all the way down the axon.